Thank you very much. It's a great pleasure to be here. I'm old friends, former colleagues. It's very exciting, and thank you very much for having me. And it's, I must say, it's delightful being in Israel this trip, working with this fantastic group, some of whom are here at the IIAS. So today I'm going to tell you about something very new in the lab, and it involves a collaboration with this very talented postdoc, Herbert Zheng Wu. I'm going to call him Herbert throughout the talk. A theoretician named Ashok Litwin Kumar, and an olfactory neuroscientist named Richard Axel. And just a quick outline of my talk is I'm going to start by motivating the problem that I want to talk about, and it's motivated in, which still is 90% of my work, which is decision making in the monkey. But, and I'm going to support this, try to turn you on to a problem that I call the circuit configuration problem, and then our first step to analyzing it in the mouse in this delayed match to sample. Okay, and if I plan things right, there should be plenty of time for discussion, but feel free to interrupt anytime you like. It's Israel, so I didn't really have to say that, but I can't believe I've gone this long already. All right, so a little bit about why I do what I do. So, you know, Plato used this expression of carving nature at its joints, and I think that the study of the neurobiology of decision making in the monkey gives us an insight. Not so much how to make criminals make better, bankers make better decisions, but rather to allow us to see what are the building blocks of normal cognition, so that if they go awry in disease, we may have ways one day of treating it better than we currently do. And so all of our work tends to be in the monkey, or most of it until recently, but I collaborate with Daphne Shohami, who is probably well known to most of you, on this animal, and of course what we'd really like to know is that, and it's a matter of a leap of faith, that the principles that emerge from studying the monkey will have applicability to cognition writ large, as expressed by this animal, us. And of course, well, not of course, but I think it's probably going to be true that to intervene, if we're so lucky that we actually are touching on carving nature at its joints, at the joints that lead to normal cognition, if those really are failure modes in a variety of diseases that affect higher brain function, which I believe they are, then we're going to intervene probably at the level of circuits and even cells and molecules. And that's going to require a level of understanding that right now is only tractable in the mouse. And so I've moved a little bit of the lab towards the mouse. A little of the lab, but a lot of my mind. Okay, so now as a way of background, let me tell you something about decision making and what motivates the problem that I'm going to share with you today. This is, we study a part of the brain in the association cortex. I should have said that these larger brains, in fact I'll go back to the slide, the larger brains was associated with a growth of the cortical mantle, most of which that enlargement in the association cortex, some of it specialized for things like language and so forth, but in general it's the stuff of thought. And to understand that, I think there's a certain property that is ubiquitous in the association cortex, and that is the operation at long time scales. And I'm going to illustrate that with the part of the brain that I study, which is in this intraparietal sulcus of the monkey, coronal section of a monkey's brain at this level. And this is the ventral division of LIP for the most part, where we find many neurons that have persistent activity, and it's of a particular kind, spatially selective persistent activity. And so if you watch this monkey, you'll see his eye positioned in yellow. If you watch him remember the location of that briefly flashed spot, here's where it was, and just waiting to make an eye movement to it, you can hear the discharge that I'll call persistent, and it'll be obvious to you why. All right, so that's the onset and offset of that spot, and this represents the go signal to the monkey, and this is when he actually began his saccadic eye movement to that remembered location. And this is what we refer to as persistent activity, that elevation and firing rate that you hear and see here by the tick marks is the time of the spikes. Now, I think that this persistence and the operation at longer time scales holds the key to many aspects of cognition and ultimately its disorders. And I refer to this as a freedom from immediacy. I forget her first name, Eva, yeah. She told me that, I got this from my inspiration, and in Eva's too, is the philosopher Merleau-Ponty. And she told me that he coined the term thickness of the present, temporal thickness. Anyway, the idea that we can process information in a time frame that isn't dictated by immediate changes in the world or the need to control body musculature in real time, I think that's the key insight in evolution, or the key change in evolution that led to what we now call cognitive function. It's pretty much a very broad term. Now, that persistent activity in LIP is spatially selective, and this is the evidence for that. If you missed it, the target was flashed to the left here, and now the activity, if anything, was depressed. It certainly wasn't elevated compared to background. Okay, so and so what we do in the lab is we contrive tasks to take advantage of neurons like this. Now, these neurons with persistent activity are all over the association cortex, okay? But we contrive tasks, they're not at all natural, and ask monkeys to make decisions between, say, a target that's in the response field of a neuron, so we expect an elevated discharge, and a target that's not. And like we might ask him to make a decision about the net direction of these dynamic dots, okay? So you should all see this net direction is to the right. If not, talk to me later. I'm a neurologist. Okay, so, but anyway, this is actually the easiest thing we ever show the monkey in our experiments. The way these dots are made is that every time a dot's plotted, a delta t, which is 40 milliseconds, a delta t later, a dot is either replotted probabilistically in motion, or with a 1 minus that probability is part of the random background field. And the probability here is 0.5, and it's the easiest thing we ever show to the monkey. I'm not going to show you data from those experiments, but I guarantee, but the monkeys in my lab are perfect, no lapses. They're just perfect at this easy. Okay. Now, if you don't really see it, don't worry. You don't have a problem. You just that they don't come out as easily on your projector. Okay. So. It turns out that, of course, it's, as I say, we contrive the task by taking neurons that have a target in their response field and ask the monkey to decide between one target that's outside of the response field and one target that's in. And so it would be trivial, essentially, to see, we expect to see, responses that are elevated before one eye movement and not the other. But in fact, the neural responses, the average being plotted here for about 55 neurons, as a function of time, the neural responses are evolving and giving us some insight into the actual computations that give rise to the decision. Okay, so what this is firing rate plotted where 0 is when the random dots were turned on. And you can see that the solid curves are ones that are going to end in the monkey choosing the target in the response field, and the dashed ones he's going to choose the other. I'll just call those right and left. Of course, they're different on different experiments because we don't know where the response field is going to be. But I'll just call them right and left. And so right away you see that these neurons predict effectively what answer the monkey's going to make. Okay, but the different colors represent the different difficulties. The gold is the one I just showed you, where 50% with probability 0.5, the dots are replotted in motion. The blue is actually, the monkey might as well be guessing, but we're mixing in all the different trials, and most of them are very difficult, so he doesn't know that he's rewarded at random, and the responses are clearly giving some indication of which way he's ultimately going to choose. And then this red is one of the intermediate motion strength, and you can see it rises and declines at an intermediate rate. So what that tells us is that we're looking at neurons that aren't just telling us which eye movement the monkey's going to make, but something about the quality of the evidence leading to the choice. And the dynamics are interesting because this is a reaction time task, and the monkey gets to decide when he's ready to give the answer. So we know that this is a period of time when he's deliberating. In fact, we've cut these curves off at the median reaction time so that you don't really see any kind of activity related to the eye movement. Anything within 100 milliseconds of the eye movement is not contributing to these averages, for example. But we can, of course, look backwards in time from the time he does make the eye movement, the saccade, the fast eye movement to one of the targets, and you see that the responses become quite stereotyped when he's going to choose the target in the response field. Not so much down here, and I'll come back to that. Okay, so what it looks like to us is that, well, let me just say that, let's look a little closer at this before I tell you the punchline, is that, instead of these 3 motion strengths, let's use all the motion strengths and group the responses by quantiles of reaction time. Now we can see that these are the fastest reaction times in gold. These are the slowest ones in indigo or whatever, dark blue. And you can see now that the responses come to a point of minimum variance about 80 to 100 milliseconds before the monkey makes his eye movement, okay? And I want to point out something else, so what we think is that that's a sign of a threshold that's been applied to these neurons or neurons like it, because this is not the only place in the brain that we see these kinds of responses, and that terminates the decision. That's how the monkey works out that he has enough information to go by. Okay, I want to point out also that there's another latency here that's quite long, about 200 milliseconds. So if we add 200 and 100, we actually account to within maybe 10 milliseconds of the amount of time that the monkey's using of the reaction time, that he's not deliberating, that he's not using evidence, okay? So I'm not going to show you all this because today's talk is not so much about this older work. It's still ongoing because we're studying things like confidence and change of mind and all kinds of aspects of the decision process. But I just to point out that it's a highly quantitative consilience between what we see at the neural level, even individual trials, and what we think is going on in the brain, which is the accumulation of noisy evidence to reach some threshold. Now, the fact that we reach a threshold when the animal chooses the target in the response field and we don't hear implies that what's really going on in the brain is a race between a mechanism that accumulates, say, right minus left, and a mechanism that accumulates left minus right. Now, if those were the same momentary evidence coming from the visual cortex, then they would be exact replicas of each other, just mirrored around the horizontal axis. But they're not exactly the same, and that's why when the monkey answers left, it's that this mechanism on the other side of the brain, for the most part, has reached its threshold. Okay? All right, so that's kind of a detail. I want to say that these neurons are not specialized for motion. They're not even specialized for decision making. And I in no way want to ever give the impression that I think that this part of the brain is a decision making area. It's a part of the brain that kind of, because it projects to the oculomotor system, in fact, I should have said, it was identified by Richard Anderson on the basis of being the part of area 7A, that Brodmann's area 7, that projects to the frontal eye field and to the superior colliculus. Okay, so what I would say is, what do these neurons care about? Using that metaphor, they care about position in space that are the goals of a possible eye movement. Okay, so we can use words like salience and attention and planning and whatever you like. I like the word provisional affordance, we're borrowing from Gibson, and maybe we'll come back to that, or maybe we won't. But you can use whatever words you like, and it's not the only reason these neurons have anything to do with decision is that we're supplying the reason for making this eye movement, and it's interesting that it shows up in the activity computationally, the accumulation, the integral dt of the evidence that leads to the choice, okay? And that would occur in the association cortex. Okay, now I wanted just to show you that that we could use different kinds of ask monkeys to do different kinds of things, and just to give you a little video anecdotal insight of a paper that's published by Shinkira and Tianming Yang. We have the monkey to make a decision right or left based on some shapes that come and go every quarter of a second. Okay, and the monkey has learned with extensive training that some of those shapes bear on the positively on the probability that the reward is at the right target, and some bear probabilistically on the reward at the left target, and all the shapes have different degrees of reliability. And what we find in the brain is that and in the behaviors, that the monkey does make his decisions based on the appropriate weighting of these cues. Okay, the appropriate thing to do is to accumulate in units of log likelihood ratio. All right, so I'm now just going to show you the same kinds of neurons selected the same way, so we assume they're the same neurons. But when a monkey is making decisions, I'm just going to give you a couple of examples. This target here is in the response field of the neuron. This one's out, so we expect this neuron to increase its discharge as the monkey is acquiring evidence in favor of the reward being here versus here. Okay, so you saw the monkey's eye position in yellow. You saw him choose the left target, which is the one in the response field. What I'm showing you here are the spikes that you heard, of course. And what I'm showing you here is on a scale, this is the log likelihood ratio, the cumulative, in favor of the target in the response field. And you can see that the first shape favored that target, and there's a little discharge, and then the second shape actually kind of reversed things so that the net, is pretty strong evidence, so the net evidence was actually for this target out here, and you can see that the response kind of dropped. Okay, I'm reading tea leaves. You shouldn't trust me on this, but lots of data analysis supports what I'm saying. And then there's mounting evidence, you see, for the target in the response field, and that's associated with a crescendo in the firing rate. Okay? Let's look at another. So here the first shape favored the target inside the response field. Sorry, I'm not reading my own axis right. Second shape added more, and the third shape went against it, but still the net evidence at this point was still in favor of the target in the response field, the one he did not choose ultimately. But then the next set of shapes overall led to mounting evidence against the target in the response field, and that means for this target, and yet that's associated with this decrescendo in the firing rate, okay? And so you hear it's kind of a cool thing to do. Here we are listening to a single neuron, in a sense chosen randomly. I mean, we go into this area, but we don't know which neuron we're going to actually encounter. And we hear this neuron doing interesting mathematics. It's adding and subtracting doing effectively the running sum of that, so a discrete approximation to integration. And it's doing it I'm not showing you the evidence for what I'm about to say, but it's doing this adding and subtracting in units of log likelihood ratio, which is the right thing to do. So you can read this paper if you want the details of all this. But my point I want to make is that these neurons in the parietal cortex somehow have access to very different kinds of sources of information to operate on in this way, in this dynamic way. Now let's go back to the random dots, and now I'll get a little bit. I'll be able to make a more specific point. So, if we train the monkey properly, or I don't know, proper or not, but if we train the monkey to handle it, he can see random dots away from the fixation point and answer left or right by making an eye movement to this target or this target, say, that happens to be in the response field. But on the next trial, we can show the monkey, if we've trained him, we can show the monkey a motion in the middle near the fixation point. For some reason, I'm not seeing the fixation point, but it doesn't matter. In the center of the screen, and now this same choice target or neurons that represent this choice are listening to random dot motion northeast versus southwest at this place in the visual field. Or in another trial, we could ask the monkey to choose up versus down based on the evidence coming from another part of the visual field. Okay. Now let's think about that problem. So here's there's an old picture from Roger Tutel from the Devoloi lab back in the day when Rafi and I were both grad students. Rafi, did you overlap? Where is Rafi? You did overlap. He was kind of a maniac, right? Yeah. Okay. His partner, what?
We published together several papers.
Oh, okay. That was the one sensible thing that he did, I think. Yeah. And he did many sensible things. But he had a partner. I wanted to go into long stories, but I almost died as a grad student because he came at me with a scapula once. Scapula? No, that's my shoulder blade. Scalpel. Yeah. Okay, yeah. I wish it would have just been a scapula. That would have been good. Not my scapula, though. Okay, so, all right. Well, anyway, what they did was, so this is just a flattened visual cortex of the monkey. This is a 2-deoxyglucose pattern of this visual field thing. And I'm just showing it to you to say, here's roughly where the choice target is represented in V1. And here's roughly the way the northeast and southwest motion is represented near the fovea. Okay, remember that was in the center of the screen. And then here are the other two stimuli. Now, somehow or another, a neuron in LIP, say, that represents this point in space, that is that choice target, has visual response often, and certainly a planning response to make an eye movement to it. Okay, again, we can use whatever words we want. It has to receive information. Right at the beginning of the trial, it's got to either get information from here or here or here, okay? And of course, those are just 3 possible locations. So that's a routing problem. And so now I've given you a couple reasons why I think there's a serious problem that we have not really worked on as well in systems neuroscience as perhaps we should have, up to this point anyway. And that is the problem of circuit configuration, dynamic changes in circuit configuration. What a cognitive scientist would call control as opposed to processing. I'm thinking of a famous essay by Norman and Chalice. Okay, so how can the same sorry, I'm getting old and I have to put on my glasses and I read. So how can the same decision or plan, proposition find support from diverse sources of information? And because propositions are entertained in persistent activity, not in neurons that keep up with the changing world or that move the body immediately. Okay, okay, good. And then a sense in which this is context dependent. How does the same information lead to one behavior or another contingent on a second contextual cue? And that's the problem that I'm going to focus on in our first attempt, anyway, in the mouse. Okay. All right. So now we're into the main substance. I have no idea how much time has passed. A lot. Okay. All right, so what we did, what Herbert Zheng Wu did, I'm going to call him Herbert, is he trained a mouse, or many mice, to do a simple olfactory delayed match to sample task. And this is a symmetric version of this task that involves licking to the left or right. The way it works is the mouse, I'm going to keep doing this, by the way, say monkey, I mean mouse for a lot of the rest of the talk. The mouse gets an odor sample. These are the names of the odors. I'm going to call them A and B. And then that sample odor is followed by a delay, typically 1.5 seconds, sometimes longer. And then he gets a test odor. And if the test odors match, then he's supposed to lick left. And if they don't match, he's supposed to lick right. So that's delayed match to sample. I want to point out that it has a certain formal similarity to a famous problem, the XOR problem, just a logic problem. So you could see the correspondence between this problem and XOR, which means that both cues are necessary in order to solve this problem, and XOR is kind of interest to computational people because it can't be solved with a simple linear classifier. Okay, but I want to encourage you to think about this problem a little bit differently. I want you to think about the sample odor as a contextual cue. If the contextual cue is A, then the mouse effectively has to use a rule between test odor and response, and that is that A means left and B means right. But if the contextual sample is B, then B means left and A means right. Okay, so we can sort of think about solving this problem as a context-dependent change in a rule. Okay, a selection of a rule, a simple sensory response association rule. Okay, in a sense, what I'd like to call it is a decision about a decision. Okay, which is a way to think about executive control or cognitive control. Okay, so here's the mouse doing the task. He's looking to the right because, and the hard thing about training the mouse to do this is it's pretty easy to get them to do this task. You should go to the left on this one because it's a match, and there he is. Okay, you're looking at him from below. You get that? Okay. The difficulty is just getting him to delay his licks until the time that the test comes. But otherwise, it's pretty easy to get him going. And we have various criteria, but ultimately, as you'll see, most of the mice perform at about 90% correct by the time we do the experiments. Okay. So very quickly, because it turns out not to be terribly interesting, but everyone's going to want to know it, we did basically a survey of the neural responses that could support the task. And so these are the connection from the main olfactory bulb to ultimately the machinery that controls the licking. And it's pretty complicated, but we selected 3 areas to study. There were good reasons for this. So the piriform cortex, which is the primary sensory cortex for olfaction, the ALM, the anterolateral motor area, which is a part of the secondary motor cortex of the mouse, and Carl Swoboda has shown over many elegant papers that there are neurons there, many neurons that essentially plan licking and other facial movements, and then in between association area, the orbital frontal cortex, OFC. Okay, so I'm just going to show you what we see there. We see many representations of odor identity, especially in piriform cortex. This is a response to the B odor. Yep, and it's a response to the B odor again in the test period. Okay, and they're so similar that I would say that's a classic sensory response without any adaptation or facilitation. Okay, yeah, sorry about that. And then we see some neurons in piriform cortex with persistent activity, so that this representation of the sample odor persists all the way until the time of the test, and you kind of might think you probably need that in order to do delayed match to sample. And so just looking across the 3 areas, This is an index for the selectivity. It's basically an ROC-based metric, okay? So no overlap at all of the responses. If it's a 1 or a -1, sign, so prefer A versus preferred B, and complete overlap if you were at 0. And what you see is that these are just the individually significant units at a like 0.01 level, and piriform is just loaded with very selective neurons, more than you might expect, given these are only 2 odors. And OFC also has many neurons like this. They tend to have a little bit more mixed selectivity. In other words, they show signs, some signs of what the sample was at the time of the test, for example. Okay, and ALM has very weak sensory responses. I'll come back to that. And of course, there's the representation of choice, which is very prominent now in ALM. Okay, so this is just a lick. You think I've got this right? A lick right cell. This is a non-match. Okay, so that's not a big surprise because again we start in the motor cortex much thinking like a monkey person, that if you want to understand cognition, don't start building forward from the sensory representation. Go backwards from what it's good for. Start with the motor system and go back to that persistent activity that supplies that is providing the reason that we do the things we do. A little philosophy for you. It's what the world of neural networks gets all wrong. Okay. Okay. So in ALM, of course, these kinds of neurons are the dominant type. And in OFC, we see neurons that have some choice selectivity, but 0.5 is roughly in this scale is kind of like D prime 1. Okay, but that's all right. And in piriform, we do see very little choice selectivity because they care about the odor, and the odors are balanced for which choice is going to be. Which choice they're going to lead to. So here's a table of the kinds of responses we see at the time of sample, at the time of late delay, because that's kind of a critical time to still remember what the sample is, and then when the test odor comes, and at time of choice. And you can kind of see this gradual change in time. I want to point out one important point, which is that if at the end of the delay, in our electrical recordings anyway, we found very few neurons that had any representation in ALM of the sample odor. And that's going to matter in a moment. Okay? Now, as I said, some neurons have sensory responses in piriform and especially in OFC that some people would call mixed selectivity, or you might say facilitation depression, or some people would call trial type selectivity. So when the test odor comes, there's a difference in the response depending on whether the first odor was the same or different. And so sometimes we record up to 100 neurons at the same time with these fancy electrodes. And if you're doing real neuroscience, according to my misguided friends, you must train a decoder to figure out what's going on. And so we did that. We wasted our time on that too. And so we found that with 50 to 100 neurons, you could decode to reasonable level of accuracy whether or not it was a match or a non-match. Okay. So keep that in mind. I'm going to come back to it and say more obnoxious things. So there's sufficient information in OFC and piriform, for that matter, to solve the task. And obviously, just with one neuron, if you choose the right ones, which is the dominant neuron type, you can tell which way the animal's going to lick in ALM. Okay, so anyway, that leads us to like the main sort of what I would call the traditional hypothesis, which is that upstream areas to ALM, that is, resolve match versus non-match, and they instruct ALM, or you could say that ALM is conceivably doing decoding like we did with our support vector machine. Okay, so so now to test that, it's really almost like a sanity check, but it turns out to be much more interesting than that. What we decided to do was do some inactivation experiments. We didn't inactivate piriform cortex, because, and I can tell you why we didn't do it. But our main focus initially was on ALM, and then I'm going to have you think of OFC as more as a control. But in any case, what we did, the critical thing here is that we inactivated by expressing channelrhodopsin in the inhibitory neurons. Okay, and so we inactivated like both sides of the brain. Okay. And we did it only during sample and delay, and then we ramp off the light so that by the time the test comes, ALM is in its normal state. And I'm not going to tell you all the controls that, I'll tell you a couple of controls, but I'm not going to tell you all the controls that support that. We think ALM is this all set, it's like the normal ALM by the time the test comes. And remember, you can't solve this task until the test comes, okay? So, all right. So here's what we see. We see a tremendous impairment in the animal's performance on the task. So this is with the laser off, this is with the laser on, and so that was pretty striking. And it shouldn't have been the case if OFC, or really any upstream area, including the ones we didn't record from, had the answer and could supply it to ALM, or even had enough of a representation so that ALM could decode it, okay? Somehow or another, by being shut off during sample and delay, we have impaired the mouse. I've probably said monkey 10 times, but I mean the mouse. Okay. And in OFC, we see, we don't really see much of an effect. Why that is, I don't know, but there are lots of areas that are upstream to ALM. But you know, we assume that AOFC plays a role, and in the literature, it's conceivable it plays a role in learning of the task. That would be compatible with previously, yeah.
Did you shut down only during the sample?
No, sample and delay.
No, only during the sample.
Yeah, I'll come to that. Well, I'll come to that. Anything in particular you want to know about that? Okay, yeah. We get less of an effect. Okay, so, but I'll show you that in a minute. Okay, so here, one of the controls that I think is worth talking about is we trained the mouse on a we call the C by CD control. And in other words, the sample is always this new odor C, and then the test is C or D. And with C or D, he's trained to go left or right. So the sample doesn't matter anymore, okay? And in that situation, the animal is shows very little impairment, basically driven by one case, okay? And these are all interleaved, and so you can see that we reproduce the original result. So this is, control laser for the AB by AB, the task we've been talking about, and this is this control task, which I shouldn't have called it control here, but whatever. But it's the C by AB task, and you can see that there's very little impairment, with the one exception. The thin lines, by the way, are individual experiments, and the thick is the average. If you haven't figured that out, sorry.
These are the same animals?
Yeah, these are no, these aren't the same animals as in the first experiment.
No,
yeah, these are randomly interleaved in the same experiment. Yeah, so that's why I think this is one of the more compelling controls. There's a bunch of other controls, but so what is it? So it tells us that ALM's playing an important role in this delayed match to sample a task, specifically under conditions when the delay is instructive. And inactivation doesn't impair the animal's capacity to translate a decision about odor into a lick response. That's one of the important things to glean from that control. And inactivation of ALM, I didn't tell you this, but there's another control, that inactivation of ALM doesn't affect the neural responses in OFC and piriform, especially at the time of test. And so we can decode just as well from piriform and OFC even when ALM is inactivated, okay? Now, I want to make another point, a little bit, get on my soapbox again, that signals upstream in all upstream areas, to ALM, don't impact. I mean, they can impact ALM at the time of the test, right? But they don't resolve match versus non-match, despite our decoding. So, I will show you this. You can keep this as a cautionary note to your colleagues, that just because the experimentalist/statistician can decode information in the brain does not mean that the brain decodes that information, okay? So we have to be careful about that. Okay, so, I try answering that to a reviewer when they ask you to train a decoder. You're better off just doing it. So everyone should learn how to do it, even though it's a complete waste of time. Okay, so, obviously decoding does tell you there's information present in a network. Assuming you don't make mistakes in your decoding, but, but anyway. So that kind of eliminates our first, what I call the standard kind of feed-forward kind of hypothesis, and forces us to consider a kind of what seemed at first glance a kind of a strange alternative, that ALM itself resolves the match versus non-match as lick left versus lick right, and it does it using information that it acquired. In the end, he has to be told what the odor is, so it gets information in the test epoch. But in some ways, it relied on some state it was in during the delay period. Okay. So the problem with that is that, as I told you, the graph I circled, we really didn't find much evidence, any really, for persistent representation of the odor in ALM. Okay. So it turns out that we probably didn't, or Herbert realized that we didn't sample the very superficial layers of cortex as well as we could have. At least they were, when we did a histogram of layers and so forth, we didn't have many neurons there. So we went back and did calcium imaging. So these are creline mice with GCamp6F, the fast calcium sensor. And what I'm showing you here are 200 neurons in a field. Okay, and so they're sorted. This is not raw calcium signal here. I'll show you that in a minute. But this is just a heat graph of the selectivity for odor from odor A versus odor B. Okay, unsigned. All right, and so about 40 neurons or so are somehow representing that at least are informative about odor A versus odor B in this field of 200 neurons. In ALM. We just missed them with the electrophysiology, and probably the reason why is they tend to be concentrated. This is the raw calcium signal now. They tend to be concentrated. No, this is not. This is still the D prime thing. They're concentrated in the first 100 microns of layer 2. Okay, so there's some scattered in, like around here. We don't know what's going on below upper layer 5 because this is as far as we image. We know we can image this far though, because what we see lick neurons in all of these layers. Okay, and even the predominant neural type, even in the first 100 microns, are lick left and lick right neurons. Okay, but these neurons seem like we missed this with the electrophysiology. And if we just look at some of them now these are the raw calcium images. Okay, so this is a couple of neurons. What did I want to tell you about these? Yeah, so they can. I'm going to show you a couple that have different temporal courses. So this is a B selective neuron with a when it responds, it tends to respond during the sample. This is an A selective neuron. This is spanning a lot of the delay. These are calcium images, and so when you deconvolve, it's a little bit less duration than this, but nonetheless, they're spanning the time. And then you have these types of neurons, which are transient. But if you look at individual trials, on average, they span the delay, but the responses come at different times. Okay, I've just stacked them so you could see the ordering. There's nothing to do with the trials. Okay, so you see what I'm talking about. So what you have in in ALM is this interesting set of neurons that do represent, in the population anyway, the representation of sample odor A versus odor B. Okay, and they come in different flavors. Some like this, some that have short durations, some that have long durations, and some that have very high scatter in when they occur. Okay, that's what this cell is. Okay. Now, we think these neurons are related to what goes on to the mouse's performance. And right now, that's pretty soft, but this is the best evidence we have. It's kind of monkey-style reasoning. So what I'm showing you here are responses of these neurons, and I've converted them to percentiles. This is for when the neuron is seeing in the sample its non-preferred, and this is when the neuron is seeing in its or smelling, so it's preferred odor during the sample. Okay, and so what I'm showing you in the heat map is the probability of an error. Okay, and so what this is the median response of say a odor of a neuron that prefers A to odor B. It's non-preferred. Okay, so that's the median, and so this is the when it happens to respond stronger to its non-preferred odor, that's when the animal is more likely to make an error. And we can do the same thing with this axis. When the neuron, this is the median response for, say, a neuron that prefers A, to odor A, it's preferred, and when it's responding weaker, that's when you see the errors, okay? So this is one of these trial-by-trial analyses. If we were in the monkey world, we would call this a choice probability, okay? And so this is as good evidence as we have today that these neurons are in some way involved with the actual task itself. In any case, they're the only candidate that we've been uncovered in ALM so far. Okay, so now what? So what's going on, right? So I want to tell you a couple of things. So there's let's see. There's two big observations I want to share with you, and that'll allow me to sort of to conclude. So the first one came up in your question, and that is that the impairment is alleviated if the inactivation doesn't span the entire sample plus delay. So here are the conditions. The control, no inactivation at all. In other words, sample and early delay, just the late delay, and then the main experiment I've been showing you all along, which is sample and delay. Okay, always turning this thing off for I think 250 milliseconds. Okay, so and so what you can see is that here's the big effect. Again, now reproduce a third time, and you can see that you do get some impairment with just inactivating in sample and early delay, but not nearly as much as you get with the whole period. Similarly, with when you inactivate in the late delay. Yeah.
So is it also the same if you change the intensity of the laser?
I don't think we've done it. Or if we did, it was like piloting and not really finding that it was a useful manipulation. So. But I'd have to check with Herbert on that. Okay, so this is now. What does this mean? I think this has some interesting implications. And so Ashok Litwin Kumar, who did a put together attractor network models, and let me just say as quickly as I possibly can. Is that what he basically it's a 2-layer model, but we're going to call it 3. Piriform, OFC, and ALM. And what he did was these curvy gray arrows mean that he trained. They're randomly connected, but he trained recurrent connections within each layer in order to solve the task. Okay, and so we tried various kinds of models, and so for example, the one I'm showing you illustrating here is one that doesn't have any recurrence in ALM. So that makes ALM a readout. And if you simulate the experiment, then what you find is that there's just no impairment at all, because that's sort of the logic of this experiment. If ALM was just reading out what was calculated upstream, then our effect should not have then our inactivation of ALM should not have impaired the mouse's performance. Okay, if he makes ALM be the only layer that has recurrence, then you should get a devastating effect. Hold on, I went one too fast. Then you should get a devastating effect, and we don't see that at all. Okay, and if you allow for some kind of persistence, that is through recurrence in ALM and the earlier areas, then you get this nice graded effect. But I want to point out one thing, is that the way the attractor works is effectively sort of in English, is that colloquially, it's that it establishes a state of the circuit that is a kind of readiness for the test odors to move into these attractor basins. It's sort of one way to think about it anyway. And so it predicts that there should be the same level of impairment for end of delay and the entire interval. In other words, if you could just interfere with the recurrence at that time, you'd be then it should mess things up. But that's not what we see in the data. So we think that we favor a different class of models. And to set that up, I just want to go back to something I told you earlier, which is I encourage you to think about this task a little bit differently than match the sample, but as a context-dependent change in the rules. And it turns out it's very hard. One clue that we have that might be correct is that if you train mice on A and B, and now you train them on 2 new odors, it takes them forever. They don't know about match and non-match. Okay, you can train them on 2 new odors, but they don't seem to generalize. But you can train them much quicker by introducing new odors at the time of the sample. Just replace A and B with these two new odors, C and D. And now, of course, we can't call this match and non-match. We really have to call it a context-dependent change. And when and animals can learn that readily. And it turns out that we now are collecting data in this task, and we know that they also involve. We don't know that ALM is causal yet. We're doing those experiments now, but we know that they involve these layer 2 neurons and so forth. Okay, so we can think about mechanisms that don't require attractor dynamics, that is persistent activity at the level of spiking, but ones that probably involve synapses. Or my favorite model is a dendritic gating model. I think the idea that maybe what these layer 2 neurons are doing, and this is just me speculating right now, is that they're turning on or off branches of dendrites, and then if the sample odor gets relayed to ALM at some point in time, then that's enough to instruct these layer 2 neurons. Let's say, again, I have no evidence that this is true, that they can then turn on or off a dendrite. And this presupposes that the test odors A and B are concentrated on different dendritic branches. Okay, we're obviously doing all those experiments to try and follow up on those predictions. But that's our working model right now. Okay, so now I'm going to close, hopefully with some time for questions and discussion. So premotor area ALM is involved in a key step in olfactory decisions, and that's not surprising from work on perceptual decision making in primates. Okay, so my view is that knowledge states live in persistent activity, as I said before, and persistent activity should be thought of more related to the motor system because of where association cortex projects downstream to things that make us do things, or I would just say entertain propositions. And so it's not, to me, it's not a big surprise that working back from the motor system, in this case being in the premotor system, is the site of a kind of cognitive operation. Okay, so upstream sensory and association areas must supply information about odor identity by the end of the delay, but they don't supply ALM with the decision. And our working hypothesis now is that ALM receives input about sample odor identity to alter effective connectivity of the lick left, lick right neurons, so that one or the other odor input at the time of test leads to excitation. And the layer 2 neurons are a promising candidate to mediate that change in circuit configuration. And so XOR, in this case, being structured as a decision about a decision, and that is implemented in the brain as a circuit configuration change in a context-dependent way. And I already alluded to the idea that maybe this is decisions about decisions, or let's say decisions about decisions about decisions. This is called strategizing and playing chess. And my view is that those mechanisms are going to force us to engage our computational neuroscience and our experimental neuroscience using language like how circuits change in order to control how other circuits work. So the outcome of a decision not being information that's for further processing, but a signal to another area or another part of the circuit that says, here's how I want you to work. Okay? Again, what cognitive scientists refer to as control. And so with that, I'll close by thanking collaborators. We had a lot of help from Carl in particular, and some of my colleagues at Columbia and Genelia Farm, and my collaborators, of course, especially Herbert, who did really all the work. And then you probably have not seen this recently discovered picture of Magritte painting his own decision process. Shall I paint ceci n'est pas un pipe or le clairvoyance? But in case you're wondering, he chose the latter. Okay, thank you.